All right, we're going to look at homeostasis. So you can refer to chapter 5 in either book. The emphasis is going to be on the plasma membrane and how the cell transports across materials across the plasma membrane. So a big question is, why are cells so small? There's a reason for cells to be so little, and I was just wondering if you could kind of think about the nature of a cell and why it would have to be microscopic. Why do they have to be so little? Well, let's think about that in terms of your life. What if your life depended upon getting from one end to the field and back again? If you had to choose between the little field and the big field, which field would you choose? I'd probably choose the little field because I want to get down there back down to one end and back to the other as quickly as possible because my life depended upon it. And guess what? Cells aren't any different. They need to get stuff in and out of the cell as fast as they can. And having a big cell means it's going to take longer to get stuff to the end zone and back. If I'm smaller, then I can get my materials in and out much quicker and more efficiently. So the size of the cell is going to be determined by the relationship of its volume, okay, the playing field, and the surface area, which is the plasma membrane. So the more membrane I have, the more ways I have to get stuff in, and if my volume is small, then there's less space for me to travel through. So cell size is going to be limited by surface area to volume ratio. So if you look at it, I have 3 cells. I have a little cell, a big cell, and a medium-sized cell. Most people tend to think go big or go home, or bigger is better. Well, not in the case of cells, because right here I have the small cell, but it has a larger surface area to volume ratio than the bigger cell. You just think, if I'm outside the cell and I have to travel into the middle, it's going to take me longer than if I'm outside this cell and I got to get in and out. I can do it much, much faster. And the membrane, the surface area, per the ratio, it's bigger. It lets me have more access to get actually into that cell. And since, like I said, if I have smaller volume, it doesn't take me long to get to the end zone where I need to go and get back out. So I always like to think of it in terms of like a balloon. The membrane is the same whether I inflate it or deflate it. The size is the same, but once I start adding volume to it, it creates space, and that space that I would have to get get maybe sugar into the cell or a waste product out. Here it's like in and out, no big deal, okay? So that's why cells are small. It's so that they can get materials in and out of the cell faster, because guess what? Their lives do depend upon it. It's not a game for them. They want to be as efficient as possible so they can get their resources in and their resources out. There are 2 basic types of cells, if we remember. There are prokaryotic cells and eukaryotic cells. And prokaryotic cells are smaller than eukaryotic cells, and that's because they lack membrane-bound organelles. Membranes allow for work. They give you a space to actually do reactions, and if I have specialized structures like the mitochondria or Golgi or the endoplasmic reticulum, I have these structures that can do specific jobs, which means I can get a little bit bigger because I have things that can do work for me. So prokaryotes, they only have ribosomes, but they're not, remember, ribosomes are not membrane bound, and they're going to be little. And that's just the nature of the difference between the 2 types of cells. Okay, so we have little prokaryotic cells, and we have our big eukaryotic cells with all the fancy membrane-bound organelles inside. So let's actually look at that plasma membrane and how we're going to get stuff in and out of the cell. So what do materials have to pass through to get in or out of the cell? Well, I just said it. It's the plasma membrane. And the plasma membrane has a tremendous amount going on for it. It's full of different types of molecules. There are protein molecules that do lots of different things. There's carbohydrate chains that kind of serve as little antennas that say, woohoo, we're a certain type of cell. And then there's this layer upon layer of stuff. And there's actually 2 layers of stuff, and this stuff is actually made out of lipids, phospholipids, and they're the part that actually makes up the true membrane, the barrier that's going to keep things from entering the cell. So there's your lipid bilayer. We got our protein molecules, and we got carbohydrates. There's also some cholesterol in there that helps give the membrane some flexibility, but we're going to look at everything as we move along. So the membrane of cells is considered to be fluid. That's what they refer to as a fluid mosaic model, because even though I have all those little phospholipids laying in there, they're kind of just like rolling around. And we'll do a bubble lab if you haven't already participated in that, where we kind of can visualize molecules moving within the membrane. It serves as an excellent model for that. But there's 2 layers to this to our membranes, and again, those molecules just kind of roll over each other. So here you can see their orientation of the plasma membrane. This is a phospholipid. It has a phosphate head and 2 little lipid tails. And the reason they orientate that way is because the phosphates like water, so they kind of stick their heads out in the direction of where the water is. And we know water and fat don't mix. So those lipids, they don't like water. They just tuck themselves in. And so we get this bilayer, this phospholipid bilayer of these molecules with these phosphate heads sticking on the outside and these lipid tails all tucked in away from the water on the inside. And it creates a really great barrier. It's going to let some things through, and it's going to keep other things out. And it separates everything that's on the inside of the cell from the outside of it. So what we call this layer, refer to this layer as is being semi-permeable. Semi-permeable means I'm going to choose what goes through. Some things can pass, some things can't. So one thing that can pass through easily is oxygen. Okay, it can diffuse right through. But a bigger molecule like sugar, no, it's not going to go through. Okay, it's selectively permeable. It's semi-permeable. Some things can pass right through, others can't. Other functions of the membrane, besides serving as a, selecting what may or may not enter it, it protects the cell. It obviously encloses the cell. It gives it a shape. And then there's some metabolic reactions that occur along inside the surface of the membrane. And then again, big role, hey, it selects what may and may not enter the cell. Those proteins, depending on the protein, have different functions. Some proteins help cells that are like in tissues stick together. Some proteins serve as identifiers so that you can say, oh, you're a liver cell or you're a heart cell. Good to know. And then some cells serve as receptor cells. So if like a hormone or some sort of chemical is released, it needs to stimulate the cell. It has a place to go to trigger the response it wants within the cell. But we're going to focus on transport proteins. These are molecules that are embedded in the plasma membrane to help move molecules across that couldn't just go through by itself. Remember, oxygen could just go straight through, but glucose couldn't. Well, your cells need glucose or they'd starve and die. So we have these transport proteins that are available to allow those other molecules get through when they're needed. So now we can get glucose into the cell. So one type of transport to get materials into the cell is passive transport. And there's lots of different examples. There's diffusion, osmosis, facilitated fusion, diffusion through ion channels, and dialysis. We're going to look at a few of these, but we're not going to hit them all. But the key thing about passive transport is that it doesn't require energy. That's why it gives the word passive. If you're passive, boom, you're lethargic, you're laying there, no energy. We're not using any energy to get these molecules across, which is great for the cell because, energy is not cheap. We want to save what we can. So looking at diffusion, we're going to move molecules across the plasma membrane from areas of high concentration, which means this is an area where I have lots of molecules, to an area of lower concentration, or the area where I don't have as many molecules. So here's an example of diffusion. When we start this, you can see all the yellow dye was on the left side. It's diffusing through the red, and it's going to do that until we achieve something called equilibrium. So why is it moving from that way, from high to low? Well, it's moving down something called a concentration gradient. And if you think of anything like rolling downhill, if you've ever been on top of the hill with your bicycle and you roll downhill, it doesn't take any energy. We're just moving from high to low, and we're doing it with the molecule's own kinetic energy, which is why we consider it passive, because I don't have to introduce energy to this system to make things happen. It's just going to naturally happen. So we're going down the concentration gradient with diffusion. We're basing it upon the molecule's own kinetic energy, and it's just, like I said, it's just going to go. It's going to keep going until it achieves that word that I said earlier, equilibrium, which means I have an equal amount at any given time of the molecule in any place. So if I start off with a high amount on the left side, eventually by the molecule's own kinetic energy, they're going to move across until there's an equal amount on both sides. Osmosis is diffusion of water molecules. But because water is awesome and it's necessary for life, we're going to call its passive transfer by its own name, osmosis. So water molecules are going to move across the membrane from areas of high concentration again to areas of low concentration. So I'm going from an area where I have lots of water molecules to an area where I have not a lot of molecules. But again, doesn't require any energy. It's just the movement of water molecules all by itself. No, and we're not referring to osmosis Jones. We're referring to the movement of water. So here you have an example of a beaker with a membrane placed in it, and you can see on side B we have our water molecules. On side A we don't have a lot of water molecules, which means we have a lot of solute, and the water is going to move through the membrane to the solute side to water it down. We're going from an area of high concentration of water to low concentration of water, high to low, until we reach equilibrium. So here's an example of that. So the left side, the orange would represent the solute, and we need to water it down. So we need to go from high water to low water. It's going to move across that membrane and water it down. And you can actually see it sometimes shift up the amount or the volume on the other side. It will actually raise up the other side. So the neat thing about water is, remember that it's a great solvent, and it loves to mix things. It loves to water things down, and that's why water can kind of be drawn into different places, because it's attracted to that solvent and it wants to water things down, which is great for your body because we're mostly water, 60% water, and our body relies on that attribute of osmosis so that we can shift fluid around where it is needed in your body. So we start talking about shifting fluid, moving water by osmosis. We need to understand some prefixes in terms of the solutions that we're going to evaluate. So hypo is a prefix that means under. Hypo rhymes with low. There's not a lot of salt. Hyper, over. Hyper, high. There's a lot of something. Iso means the same, and tonic means salt. So we're going to put these words together where we have hypotonic, hypertonic, and isotonic. And each of those are just referring to the concentration of salt in that solution. So looking at hypotonic solution, hypotonic would mean I have low salt, okay? Which means there's more water. And if there's more water in the solution, that means there's less water in the cell. And so water is going to move from a hypotonic solution into cells, and it's going to make them get bigger. And sometimes if that happens, they will actually blow up, which is sad for the cell if they don't want to be blown up. Hypertonic means there's a lot of salt, which means there's not a lot of water. And therefore there's going to be a lot of water in the cell. So the water in the cell feels like it has to water down the stuff outside of it. So the water in the cell will leave and go outside the cell, causing the cell to shrivel up and become flattened, which is no good for the cell at all. Isotonic means I have equal amounts of water and salt on both sides of the membrane. And so the cell is not going to change. It's going to be completely the same. A good example of this is individuals who have to wear contacts. They have to use a solution to clean off their contacts and then put it in their eyeball. And that solution is saline. Well, it's the same exact concentration as the cells in your body. And they make it that way so they don't hurt your eyeballs, because we wouldn't want them to blow up because of a hypotonic solution, and we wouldn't want them to shrivel up because it's a hypertonic solution. We don't want your eyeball cells to do that. We want them to be safe. So we just use that nice isotonic solution so that we can use our contacts and not hurt our eyes. So here's kind of a great example of what it looks like when we put animal cells, like a red blood cell, into different types of solution. I like to remember it by rhyming. So hypo means low. We're going to blow up your cell. Iso, it's all good. Iso the same, okay? Water in, water out, it's all equal. Hyper, I always envision something like a sugar crash. You're just losing it and you're going to crash so that it shrivels up. But this is kind of one way I remember it. Hypo means I have no salt out here, salt's inside, water's going to go in there, blow it up. Hyper means lots of salt out here, not in the cell, so we're going to go outside the cell to water it down, and therefore we shrivel it up. Iso, we're happy. We're all good. Now cells have to deal with osmosis because, again, typically your cytoplasm is going to be have a greater concentration of salt than water, so water is going to want to go in there and blow it up because water is hypotonic traditionally. So some cells have little organelles called contractile vacuoles that act like sump pumps and are constantly pumping out water so that they don't blow up, and that's how they maintain homeostasis. Plants feel a little bit different about water and the different types of solutions. They enjoy hypotonic solutions. In fact, it creates something in the cell called trigger pressure, where that large central vacuole fills up with water, but because it has that rigid cell wall, the plant cell won't blow up. The plant cell actually gets held in place, and it provides some structure for the plant. That's why a healthy water plant looks full and beautiful. If we don't water the plant, or we use a hypertonic solution that would pull water out of the plant, it loses that trigger pressure and it wilts, and that's called plasmolysis. So plants actually like that hypotonic state. So they have trigger pressure. They don't like anything else because it makes the plant flaccid and it gives you an unhappy plant. If the cell does blow up, we call that cytolysis. Remember, lysis means to break, cyte means cell. Well, we just blew up our cell. So here's a comparison between an animal cell, a human red blood cell, and a plant cell. In an isotonic solution, you can see for a plant, an animal, we're happy. It doesn't hurt our cells, nothing's changing, water's in, water's out, it's equal, we're good. The plant cell, that central vacuole isn't as full as it could be, and the plant will be flaccid and very unhappy. In a hypotonic solution, just remember, because our cytoplasm has more salt, the water will flow into the cell. Well, for us, the red blood cell is going to burst. There's nothing to restrict that from happening. Water is going to flow in. We have no way to compensate. Our cell is going to blow up. The plant cell loves it because it has that cell wall around it to kind of give it like a little structure or safe zone to work in, and it fills it up, and that's where we get that trigger pressure that plants love so that they can be full and beautiful and healthy. Nobody likes hypertonic solutions, okay? Because all the water is leaving the cell, there it goes, and our blood cells shrivel up, and the plant cells shrivel up. They actually plasmolyze. They, boom, done. So nobody likes hypertonic solutions. But trigger pressure is good. Our next example of diffusion is the facilitated diffusion, and that's where we're going to use those transport proteins within the plasma membrane to get molecules across. Oxygen, remember, can just go whoop right through, but something like glucose can't. It needs help. So we need some transport proteins to carry those molecules across the membrane because they're not going to get through that bilayer by themselves. So an ion channel will get some like ions across, like chlorine, potassium, sodium. They're like a door, and they're specific for each ion. So like a sodium ion can't go through a potassium ion channel, or vice versa. They're specific, which is fine. Carrier proteins are specific to the molecules that they allow to enter, and they will allow something like glucose to fit through and make their way through. Now we're going to look at the opposite of passive transport. We're going to get things through the plasma membrane, but it's going to take energy, and that's active transport. So a passive transport is you sitting on your bicycle and just letting it coast downhill. You're going down the concentration gradient. Well, now we're going up the concentration gradient, and that takes energy to pedal uphill. So we're in the reverse now. We're going from areas where there's not a lot of a molecule to areas where there is a lot of the molecule, okay? We're going opposite. So we need energy, and the molecule that's going to help us do that is ATP, so that we can make it up that hill and move things up against the concentration gradient. So an example of that is the sodium-potassium pump. Your neuron and the membrane of your neurons use the sodium-potassium pump to send the nerve impulse along it. This is how your body works. But it's loaded on their sodium-potassium ions are on opposite side, but we got to push those across, and then we have to reset that, and that takes that reset to get those ions back on the proper side. We have to use active transport, and that does take energy to do. Now there's a couple of more ways that we need to use energy to get things in and out of the cell, and that's where we're looking at the movement in vesicles. Remember, a vesicle is like a little package of proteins or something that the cell has made, or we need to get into the cell for the cell to use, and that's going to take some energy. So some prefixes, suffix to look at. Endo means into. Just listen to the word endo. Endo, okay? Into. Exo means to exit. Just look at it, EX. It looks like an exit sign all by itself. And cytosis means the cell. Cyte means cell. So endocytosis means we're going to move stuff into the cell. Exocytosis means we're going to get things out of the cell. So endocytosis, active transport, where we have to move something into the cell with the use of energy. And what happens is there might be a particle or something outside the cell we want, and the cell is going to come in contact with that particle, and then it's going to wrap the membrane around it, pinch it off, and then let it pull on through into the cell. So now you have this membrane-bound package inside the cell, and it will go to the organelle that it wants. Maybe it goes to a lysosome, and those membranes they merge together, and then the lysosome can break down whatever inside that vesicle. There's two different types of endocytosis. One of them is pinocytosis, which is like intaking of fluids. And there's phagocytosis, which is more like eating food. So here is endocytosis, where you can see here's our particle, the membrane pinches around, it comes inside, and here's actually, this is a white blood cell, and it would go after that bacteria right there. These are all red blood cells, and it would hunt it down and it would engulf it pretty much the same way that we see over here. But white blood cells are referred to as phagocytes because literally they hunt down the bacteria and then they gobble it up. Sometimes, though, and if we remember back, that things that are gobbled up don't die within the cell. A long time ago, we had a small little bacteria that got eaten by another bacteria, but for whatever reason, it didn't get broken down inside the big cell. It's stuck in there now. And it became an advantage for that big bacteria to have that little bacteria living inside it, because that little bacteria can make energy for the big one. Now, over many generations, billions of years, that bacteria stopped living, but it still existed inside the big one. It just turned into the organelle that we know as the mitochondria. And we know this occurred because we have evidence of that. We know that the mitochondria has its own DNA, and if something has its own DNA, it had to have been a living cell at one point. The opposite of endocytosis then is exocytosis. So I need to get things out of the cell. Well, remember your endoplasmic reticulum is making proteins. It's sending those to the Golgi apparatus to be packaged into vesicles and then shipped out of the cell. So stuff from the Golgi has to get out. So these vesicles from the Golgi are wrapped up in a membrane. They're going to go to the plasma membrane, and again they're going to merge together. And as they merge together, the stuff inside gets expelled out. Now, it could be something like a protein, or it could be a waste product or toxin that we need to just, say goodbye to. But remember, this is because, so this stuff is coming from the Golgi apparatus. So that's it for homeostasis, cell transport, and the plasma membrane. Again, refer to chapter 5.